This video, what we're going to do is perform the experiment in the Enzyme Lab. Over here on the left are our procedures in one document, and on the right hand side is our actual lab simulator. So to begin, what we'll do is we'll start the lab and make sure that you have your PPEs or personal protective equipment and enter the lab. And what we're going to do is for our first experiment, we'll take a constant temperature bath, which is right here, put on our bench a thermometer and a gas syringe here. And then it says set the temperature bath to 10 degrees Celsius, so we'll just click the left button and that will put it at 10 degrees. And then take a small test tube from the container's shelf and place it in the constant temperature bath. So that's right here. And we'll just drag that and place that there. Now it says add 5 milliliters of 3% hydrogen peroxide from the materials shelf. And this is our 3% hydrogen peroxide. You drag and drop, and then you can enter the amount 5 and add. And then you can see it fill up. Then we're going to add 5 milliliters of water to the test tube. So our water is right here. We drag that. We'll add 5 and add. And you can see the volume goes up again. Now it says move the thermometer from the workbench to the test tube. Here we go. And see, it's a fluctuating reading because it's theoretically supposed to be reading this real time. And it says wait until the temperature stabilizes at the same temperature as the bath. And so we may wait for a few minutes. And what we might do is see how this is Celsius and this is Fahrenheit. You can click that to change it to Celsius so that it will read the same temperature scale for you. And so as this gets close to 10 degrees Celsius, we're going to think about or read about what's the next thing we're going to do. It says move or click move the gas syringe from the workbench to the test tube, and then click the gas syringe and note the initial volume of the gas. So it is always a good idea to kind of read the next step before you do the step you're on, just so that you'll have an idea of what you're doing. So we're going to move the gas syringe and then click it to make an initial note of the reading. And so we'll move this here. Oh, try that one more time. And then we'll click it and see the volume says 0. And so that's what we want to read. And then it says add 1 milliliter of catalase solution from the material shelf to the test tube and immediately note the time on the lab clock, particularly the seconds. The lab clock is down here, so you're going to want to monitor for approximately 15 seconds because the next step says after 15 seconds elapse, click the gas syringe again to determine the volume of the gas released from the reaction and record the two volumes. Your initial, which we already know is 0, and then after 15 seconds. And you're going to keep those just kind of written down on a piece of paper somewhere. So let's do that. We're going to take our catalase, which we'll scroll over. I think this is your catalase here. We're going to move this to the test tube. We're going to add 1 milliliter, and then we're going to note at 37 seconds. So we want to go to 52 seconds for 15. And we're going to wait. And we're waiting. And now we're going to double click and it says 1 milliliter. All right. And so now it says move the thermometer and gas syringe to the workbench. And empty the test tube in the waste bin and place it in the sink. So we're going to empty it, waste bin in the sink. Now what we're going to do is we're going to repeat steps 2 through 13 4 additional times using the temperatures of 21.5 degrees Celsius, 40, 60, and 80 degrees Celsius instead of 10. You're going to record the optimum temperature for catalase activity on your results. And so essentially what you're going to do is record the initial temperature and, your initial volume and your final volume after the 15 seconds each time. And you're going to determine which one produces the most gas. And then you can go on to step 2 or experiment 2. So let's try this one more time. Let's move up to 21.5. We're going to do a test tube. We're going to put 5 mils of peroxide, 5 mils of water, and then we put our thermometer. And again, let's change that to Celsius. Oh, it says 21.5, so that's good. So now we're going to put our gas syringe on here. We're going to click it. Volume of 1.04, so that's all right. We'll write that down. And now let's add our 1 mil of catalase and wait the 15 seconds. Oops, it didn't work. Okay, so 26, so we want to go to 41. Double click, we have 8.91 mils. All right, we're done on that one. So what we can do now is remove these two, waste. All right, we can get our start over for temperature 40. We want to do 5 mils of peroxide, 5 mils of water, thermometer, put that in Celsius, let it go to 40 degrees Celsius. Once it reaches 40 degrees Celsius, we're going to move our gas syringe to the test tube and take our initial reading. We're almost there. Okay, I'm going to go ahead and go since we're pretty close. We'll take our initial reading is 12.79 milliliters. And now what we're going to do is we're going to add our 1 mil of catalase and wait our 15 seconds. Starting at 53. So an 08, double click, 33.65. Okay, done. Remove, remove, dump in the waste, put in the sink. New test tube. We're going to go to 60. We're going to put our 5 mils of 3% peroxide, 5 mils of water, thermometer, put that in Celsius, and we need for it to stabilize at 60. So I'm going to pause, and when it hits 60, I'll come right back. So the temperature has risen to 59.7, and it's getting very close to 60. So what I'm going to do is get ready to move my syringe. Okay. So I'm going to go ahead and go, since it seems to not go to 60. So I'm going to put that syringe, I'm going to take my initial reading, 44.46 milliliters. And what I'm going to do now is add my 1 milliliter of catalase. And then get ready to count 15 seconds. Ready, go. So we're at 35, we're going to go to 50. We look 49.78. And it's still going to be moving, but you want to take that reading at that 15-second mark. So that's okay. So we're done with that experiment. Now we're going to get rid of the waste, get rid of the test tube, do a new test tube. But this time we want to do our last temperature, and that's 80. So we're going to put our 5 mils of peroxide, our 5 mils of water, add our thermometer, make sure it's Celsius reading, it's going to go to 80. I'll come back as soon as we get close to that temperature mark. Okay, we're getting close to being at the 80 mark, so what we're going to do is make sure that we have our syringe ready to go. And as soon as we hit 80, we'll put our syringe on our test tube. Okay, well, we're going to go ahead and do it. And even at 79.9, we're going to take our initial reading, 56.06. And now what we're going to do is we're going to add our 1 ml of catalase and wait the 15 seconds, okay? 1 ml, go. We're at 20, so we'll go to 35. Ready, go. 56.84. 56.84 milliliters. Done. So now we're done with that particular part of the performing of the experiment. What we want to do now is be able to compile all of our data. And if you've been writing this down, you should have something that looks like the following. So for 10 degrees Celsius, your initial reading was 0.00 milliliters. Your final reading after the 15 seconds was 1.00 milliliters. For 21.5 degrees Celsius, your initial reading was 8.91 milliliters, and your final reading was 33.65 milliliters. At 40 degrees Celsius, your initial reading was 12.79 milliliters, and your final was 33. Let's see. Oh, you know what? I think I gave you the wrong final reading for our 21.5 degrees. It should actually be 8.91 mils, and the initial reading for 21.5 was 1.04. My apologies on that. 1.04. So for 60 degrees, your initial reading is 44.46 milliliters, and your final reading is 49.78 milliliters. And at 80 degrees Celsius, the initial reading was 6.06 milliliters, and your final was 56.84 milliliters. So in order for you to determine which one of these temperatures was the best temperature for your enzyme to react, essentially what you want to know is which one had the most volume produced. So what you want to do is figure out what is the difference or change in volume. And so really quickly on your calculator, figure out what the change in volume from your initial to final. And the way that you would want to do this in your calculator is take your final temperature, subtract your initial temperature, and this will give you your change in volume. I'm sorry, your final volume. Final volume and final initial volume, and that'll give you your change in volume. So for our first one, the change is 1.00 milliliters. For 21.5, the final volume is 8.91, subtracting 1.04. That gives you a final change in volume of 7.87 milliliters. At 40 degrees with 33.65 minus 12.79, that gives you a change in volume of 20.86 milliliters. At 60 degrees, you have 49.78 minus 44.46. That's 5.32 milliliters. And the 80 degrees is 56.84 minus 56.06, which is 0.78 milliliters. So the temperature which gave you the greatest volume, aka the most active catalase, is going to be at 40 degrees Celsius. So this is how you perform experiment 1 in the Enzyme Lab, the effect of temperature on catalase activity.